DNTOX.
Thank you very much. Good afternoon. First of all, thank you that we have the opportunity to present our idea here at the BioRiver Boost. We are DNTOX, and our goal are safe chemicals for a healthy brain development. Already during pregnancy, children are exposed to a variety of chemicals. One examples are, for example, pesticides on our food items. And these chemicals can disturb brain development and cause developmental neurotoxicity, short DNT. Consequences of chemical interference in brain development are developmental disorders. For example, ADHD, autism, learning deficits, but also loss of IQ up to mental retardation. Within the United States, every 6th child is diagnosed with a developmental disorder. However, for the majority of chemicals registered globally and within the EU, it is not known whether they influence brain development. Until now, only 140 have been tested regarding DNT. And the obvious question here is now, why are not more chemicals tested? And the reason is that the only available method here are animal experiments. And for testing of a single chemical, 1,000 animals are needed. So therefore, testing of all chemicals is on the one hand highly unethical and also economically not justifiable. And even if a human is not a 70-kilogram rat, so there are market species differences, and the transfer of the test results from animals to humans is highly controversial. We from DNTOX, we developed an in vitro testing battery to solve exactly this problem. Our battery identifies DNT chemicals animal-free by using human cell sources. The battery is already in part automated, and we thrive at full automatization. And our data analysis is supported by an in-house developed software using artificial intelligence and own algorithms. Our testing battery is composed of different individual assays, which in their entirety recapitulate the processes of human brain development. And for that, we use different cell models. On the one hand, human neurospheres. And with these, we can analyze the migration of neural progenitors and the formation of the different cell types of the brain. And here in this little example, I hope it pops up and it plays, yes. Here you can see clearly by eye how a DNT chemical in the right video prevents that the neural progenitor cells migrate out of the sphere. In the left video, you can see in the controlled condition, nice migration. And also here in the pictures, you can see how a DNT chemical disturbs the formation of the different cell types of the brain. What is important here that all these effects you can already see by eye just looking through a microscope. Our second cell model are human-induced pluripotent stem cells. And with these cells, we can analyze the electrical activity of neurons. And also here you can see how a DNT chemical clearly reduces the electrical activity. To give you a small peek into our black box, this is the full setup of the battery. And what we developed within 15 years, I cannot explain just in a few sentences. So what I want you to focus on is just the last part on the end, where the DNT battery can clearly discriminate between a DNT negative and a DNT positive chemical. Looking at the USPs of our testing battery, we test animal-free and we are therefore sustainable. By using human cell sources, we do not have to consider species differences. We are able to deliver fast results and therefore accelerate the admission process and reduce costs. Due to a lot of collaborations, our battery is already internationally recognized, and testing in our battery increases the trust in our customers' products. And my colleague Christina Bartmann is now going to present the business model.
DNTOX is a bioanalytical screening laboratory which offers animal-free DNT testing of chemicals. We support our customers during the identification of safe lead structures. And furthermore, we support the safety assessment for a chemical approval and also reapproval. We offer a comprehensive testing and analysis within our battery for regulatory purposes for €40,000 per chemical. Our financial requirements add up to around 1.3 million euro within the next 2 years. And with these, we want to perform a lab-to-lab validation of our testing methods, expand our testing laboratories, and also prepare for our market entry. Regulators are at the moment aiming to make the testing within our battery mandatory, and this is why we expect a clear scale-up within the next years. We could already acquire the first customers for our venture. For the EFSA, we successfully tested 120 chemicals between the years 2019 and 2020, and this was a really big milestone in the field of DNT in vitro testing. With the NIH, we made a 5-year contract, which includes the testing of 115 chemicals within the first year and the testing of additional up to 200 compounds. And as these are 2 pilot customers, they are not included in our sales forecast. So, in addition to the already named authorities, our customers are the chemical industry, the pharmaceutical industry, as well as different CROs. The market is defined by the number of listed chemicals. It is assumed that of the globally registered chemicals, around 100,000 are relevant for the human society, and currently about 1,500 pharmaceuticals are approved. And of course, pregnant women are also exposed to the majority of these chemicals. Currently, there is no other alternative to animal experiments regarding DNT. So we expect that leading CROs and in vitro startups may try to enter the market, but currently there is no hint that they also meet the required methodological competence. So, however, with one of these scale-ups, we are currently negotiating a future cooperation to join forces. But in comparison to our competitors, DNTOX has an excellent methodological competence in combination with our in-house developed AI-based software. And furthermore, of course, we benefit from many years of collaboration with international authorities. We envision the launch of our company within the next months. We are currently working on the expansion of our screening laboratory, as well as the further automatization of the testing battery to also meet the future testing demand, which of course increases. And for a full regulatory acceptance, we aim to also expand our quality management control system. Our team comprises 3 researchers, Katharina Koch, Stefan Macias-Hussmann, and myself, Christina Bartmann. And we are all currently working as scientists at the Leibniz Research Institute for Environmental Medicine in Düsseldorf. The foundation stone for DNTOX was laid by the year-long research of Professor Ellen Fritsche. She is leading the working group Sphere Biology and Risk Assessment at the IOF and will be our scientific advisor. Our economic advisor will be Silke Boucan, and in all bioinformatical questions, we will be counseled by Professor Axel Mosig. And with this team and our product, we want to realize our company vision, safe chemicals without animal experiments. Yeah, thank you all for your attention, and now, of course, we are very happy to answer all your questions.
Yeah, thank you.